So welcome everyone to the Spotlight poster session for the Pharmacogenomics and Personalized Medicine SIG. I think first, Jill and I will just introduce ourselves. So I'm Leah Sansbury, one of the co-chairs of the Genomics SIG. I'm a senior director of epidemiology at Ultragenyx Pharmaceuticals. I recently made a career change. Prior to that, I was in the epidemiology group at GSK and have been a member or a co-chair of the SIG for many, many years and have participated in the pharmacogenomics skill education session as well for years. So welcome. And Jill, do you want to introduce yourself?
Sure, I'm Jillian Bartlett Espelant, and I also did a recent career change. I was previously a professor, tenured professor at McGill in the Department of Family Medicine, pharmacoepidemiologist by training, and I have recently become the Associate Dean for Population Health and Outcomes Research at the University of Missouri in Columbia, Missouri, where I'm still a tenured professor in family and community medicine, but also co-director of a translational biosciences PhD, and they have a really interesting next gen initiative there, so I can indulge in my genomics passion that goes alongside my interest in pharmacoepidemiology. So Leah and I co-chair the special interest group, if you're wondering what SIG stood for, and I'm also an ISPE fellow. And we have a very active group. And normally we would be so delighted to be standing next to your poster and hearing what you had to say about this. But we are very happy to have see your posters today. And we will talk at the end of the presentations a little bit about some of the SIG's activities that we're planning for the year and hoping very much we see you in person next year. So Leah, did you want to get us started?
Sure thing. So here is the schedule. So we have the order, I think the speakers know, Sammy, Jeffrey, I don't know if we have It looks like yes, we do, and Jessica. So I will stop sharing and turn it over to Sammy to present.
And just a reminder, these are 5 minutes. I mean, we don't have a lot of posters. You technically are supposed to take 3 minutes to present. It's 2 minutes for question and answers. Might be a little more easygoing about the Q&A session, but try and keep, to be fair to everybody, try and keep your presentation to 3 minutes.
Can you guys see my screen?
Yes, we can.
Yeah, looks great.
Okay. So thanks for the opportunity. I'm Sammy. I'm a data scientist at Genentech Roche, presenting on behalf of my co-authors who are also at Genentech Roche. As hopefully most folks here know, the increasing use of comprehensive genomic profiling, or CGP, in oncology has enabled clinical genomic databases, or CGDBs, to connect patient-level data from EHRs to CGP results. However, biomarker data from EHRs and CGP can vary in assay type, specimen type, and timing of testing. The objective of this work was to quantify concordance of biomarker status between EHR and CGP data in a representative CGDB. In this work, we used the Flatiron Health and Foundation Medicine, or FMI, CGDB, which links Flatiron Health oncology EHR data, including abstracted biomarker results, with FMI CGP results. Of note, FMI CGP results are removed from the Flatiron EHR in this database. We focused on biomarkers included in the NCCN guidelines for 5 tumor types: ALK, BRAF, EGFR, KRAS, and ROS1 in non-small cell lung cancer, HER2 in breast and gastric cancer, BRAF, KIT, and NRAS in melanoma, and then BRAF, KRAS, and MMR, MSI, and NRAS in colorectal cancer. For each patient, we paired biomarker results from the EHR and CGP using the closest possible specimen collection dates. Following FDA guidance, and there's a link to that particular guidance there on that slide and the poster as well. We calculated measures of agreement using overall positive and negative percent agreement, or OPA, PPA, and NPA. We found OPA to be consistently high, generally above 90% across all biomarkers and tumor types, as you can see in this figure. And then given the generally low prevalence of the evaluated biomarkers in panel A, NPA, as you can see in panel B, was also consistently high, irrespective of the direction of the reference, and drove the high OPA. In contrast, as displayed in panel C, the PPA was far more variable across biomarkers and tumor types, again, irrespective of whether the EHR or CGP was chosen as the reference. Some interesting insights into this PPA heterogeneity were provided by sensitivity analyses evaluating the impact of genomic variant type, the sample type, the test type, and the bait set type that was used in the CGP analysis. So in conclusion, while the high proportion of biomarker negative patients drove high OPA and NPA between the EHR and CGP data in this particular database, the PPA was heterogeneous, depending on technical differences between assays, the direction of the comparison, the biomarker itself, and the tumor type. Deciding whether to use EHR or CGP data as the biomarker source in light of these PPA discordances thus requires careful consideration of these factors, and also importantly of the specific research hypothesis that's being probed. Our hope is that this work provides a set of helpful quantified reference points for future studies using biomarker data in CGDBs. And finally, thank you for the opportunity to share. These were the slides. I can project the poster if more useful for discussion. Just let me know.
How about for the question period, you pull up your poster quickly?
Sure, sounds good.
Okay, this is a really silly question, and then I'll entertain more serious questions from our participants. Flatiron, where is that?
Where are they based?
Yeah.
They're based, they started in New York City, in the sort of Flatiron district, I guess, of New York City. But they have an outpost in San Francisco now as well, but they're primarily based in New York City, yeah.
Okay, great, thank you. That I'm just I'm asking because I'm being from Columbia, Missouri, and we have a flat branch creek, and I'm like, so I was wondering where that came from. Okay, questions. Is any, you can raise your hand under reactions, or you can okay. Any legitimate questions for Sammy?
All questions are legitimate, right?
No, mine was just, well, maybe legitimate, but very frivolous. So I will, Leah, do you have a question?
So I was going, it was a lovely poster.
Thank you.
It was very straightforward. I'm trying to think. I mean, I think part of it too is I'm always interested, in some of the SIGs meetings that we have and presentations, what next? So you've linked this, you have the data, but next steps, and as far as
great question. And I'll be honest that part of this work, as you can guess, this was basically to serve as a reference point for us internally as well. Some of the key take homes, like in figure 2, it's nice to see it backed up by some of these evaluations, but it's almost like, okay, that kind of makes sense, right? And so I think like the really actionable stuff is really around the sensitivity analyses. And some of those examples are provided in figure 3, right? So if you're someone who works on breast cancer and you're really interested in HER2 testing and how HER2 testing via IHC, via NGS, via ISH differs, and then that figure, the data that is in that figure, is really useful, right? To be aware of in designing your study. And again, depending on your research hypothesis, like is your research hypothesis related to like the biological significance of some of these genomic changes, or is it more related to like assessing sort of clinical practice of testing, right? And so something that my co-authors and I discussed is the value of potentially following this up with a publication that might also make a dashboard available with the full granularity of the analyses, right? So that someone could access it and drill down into their specific biomarker of interest and their particular indication of interest and see the granularity of all the sensitivity analyses around it. We don't have that set up right now, but I would guess that if we get pinged by a lot of interested folks, either internally or externally also, with that kind of an interest, then that might be a driver for that. Does that help?
I think this is fabulous. I think, yeah, no, and I saw Donna hopped on too. I'm not going to make Donna ask questions, but my previous life, I was in, I supported the oncology pipeline, GSK, worked with Flatiron. We had a workshop this morning on external controls using Flatiron data, but I think specifically like this, the biomarker aspect is one that's, I don't think it's sort of underutilized. And I think this is it, but lots of people are thinking about it and how useful is, the Flatiron data for answering some of these questions. Donna said she put a question in the chat, sorry. So Donna is asking about, can you talk about if you evaluate patients that are testing available versus those who don't have testing available and potential biases, whether or not you can get at that information or not.
Yeah, and that's another sort of like to-do item for us that, we approach this first step in this work in a broad way across all the indications. So something that we discussed is basically in a tumor type specific fashion, do exactly that, sort of assess either in terms of baseline characteristics or outcomes or both, what potential biases might be associated with using either EHR-based biomarkers only or CGP-based biomarkers only. And so on. So lots of stones to be turned over, and unfortunately, it's not like the message is a single simple message. The message is, as it often is that it's nuanced, and it really depends on your research question, right? But yeah,
Exactly.
That was one of our next steps of evaluation in furthering this work. We're not there yet, though.
No, great. Please keep at it for those of us that are definitely interested in, the genomic and biomarker data and how to use it. And, I think the sensitive, I really enjoy figure 3. So it'd be great to see how you, if possible, making this into a tool and sharing with others as well.
Thanks. We'll convey that.
Yes, I'd highly encourage you in that. We have a next gen initiative at the University of Missouri, and being in population health, this is sort of the exact information I'm looking for people to create. And it's amazing how often there's a disconnect. Like you can get all the EHR information, or you can get the GWAS information, or the different genomics information, and combining them, where you really need a lot of different information to look at risk prediction and outcomes and everything else. This is a unique data source, and please do continue to pursue this and publish on it And the idea of a dashboard is really, really quite helpful. I am going to mention here something that Leah and I started last year and was quite interesting, is that we did ask all, because we liked all of the posters in the SIG and the poster walk, and we wanted to hear more about it. So one of the things, if you're interested, please, you can message either Leah or myself in the chat. If you'd be interested, we host a series of webinars for our SIG, and we ask our poster walk authors to give a longer presentation. So 20 to 30 minutes with a 15-minute moderated Q&A with the different people involved in the SIG. That's open, by the way, to the whole ISPE membership and anybody actually that you want to open it up to attend. So it'd be great to hear a little more than what you can convey in 3 minutes. So thank you very much, Emily. That was very interesting.
I think next up is Jeffrey.
Great. Can you see my poster?
Yes, we can.
Perfect. So thank you very much for the invitation to present this. And by way of context, I'm in rheumatology and a practicing rheumatologist as well as pharmacoepidemiologist. So I need to give you about 60 seconds of background. So rheumatoid arthritis is kind of one of our prototypical autoimmune systemic inflammatory disease, carries with it an excess cardiovascular mortality of roughly 50%. Most of the existing cardiovascular risk calculators out there make no allowance for any systemic inflammatory condition, be it rheumatoid arthritis or lupus or anything else. But inflammation is clearly a driver of excess CVD risk and MACE events, as probably all of us are aware. So what our field currently enjoins primary care doctors, rheumatology, cardiology to do is just go take any of the pooled cohort risk equation or your favorite CVD risk calculator, multiply by 1.5 simply because you have the disease, but that absolutely doesn't reflect the contribution related to how active that disease is. So the purpose of this analysis was to take a biomarker-based disease activity measure, the so-called MBDA test, or it goes by the brand name Vectra. This is a commercially available test that doctors can and do use to measure RA disease activity, and we're using it for something quite different than it's originally intended to do, which was to predict cardiovascular disease event risk for the next 3 years. This is already published, it's already available, and it's currently being used. The limitation, though, in what we did in that prior work that I alluded to in this first bullet, we did all of that work by linking the Vectra lab results to Medicare administrative claims data. So the limitation maybe is a little bit self-evident in that Medicare tends to be older people. In any biologic using cohort, about 20% of people are younger, but they're younger and they're generally disabled, and that's why they qualify for Medicare. So the purpose of this analysis is to say, take that same algorithm, take those same biomarkers, and look at it in a younger population where we're linking not to Medicare fee-for-service data, but rather to a different commercial claims data set. This cardiovascular risk score combines the multi-biomarker score, some specific biomarkers people may recognize leptin as one of the adipokines, TNFR1, as well as matrix metalloproteinase 3, and then the other 9 biomarkers. And then we include age and 4 traditional risk factors. These are ones that right now, when doctors order the test, most of them check a box on a test requisition form. But in fact, these mostly could come out of an automated data system, a data warehouse, an electronic health record. So the purpose of this is to validate using a different data set that contains a lot of younger people, this biomarker-based CVD risk score. So we use the Symphony Health Integrated Database to link the medical claims from inpatient and outpatients to the biomarker lab test results, which we obtained and have from the manufacturer. We used a one-way hashed with tokenized data to do that linkage so we don't have to exchange unidentifiable information. And then we used Cook proportional hazard model to evaluate the biomarker-based risk score to evaluate MACE event, which was hospitalized MI or stroke. And then the key part of the analysis is to do that in certain patient subgroups, because we get asked all the time, you know, great that this works overall, but does it work in people, you know, with high or low disease activity? Does it work in the subgroup of people, you know, with and without hypertension or smoking or diabetes or any of the risk factors, really to speak to generalizability. So there's the generalizability issue for younger patients in a different data set, but then there's also the generalizability of all of these subgroups. And in the original work, the hazard ratio of the biomarker-based risk score was 4. And so that is essentially where we're going to drop the dotted line in this forest plot to both look overall, but then these key subgroups. So the subgroups here are looking at the performance of this risk score and the hazard ratios associated with it in the subgroups defined by high C-reactive protein, that would be more active disease, moderate or low, the biomarker category, which also maps to RA disease activity, and then a number of the other traditional risk factors, smoking, diabetes, age, sex, et cetera, et cetera. And then all patients, and overall, this was quite similar to people who were in the database. When we look at the characteristics of patients, as we intended and hoped for, the mean age is closer to what we would expect for a more typical rheumatoid arthritis cohort, and the prevalence of various comorbidities was modestly lower than we saw in the older Medicare patient population. So bottom line is that the fact that just about all of these line up quite nicely on the referent line based on our prior work with that quite similar hazard ratio was quite reassuring and spoke to generalizability. And the fact that this is an independent data set, again, provides further evidence for the validity of this and good generalizability.
Thank you. That was excellent and very clear. Yeah, I like your chart with all the hazard ratios. So I'm going to steal Leah's question, and then she can think about her own. I am an implementation scientist in addition to being a pharmacoepidemiologist. So I also always have that question: how would you see this being used in clinical practice?
Well, and I kind of foreshadowed it a little bit. You can order this as part of a test requisition form where you check a box, but the downside is that that's a manual process. And so you might think, well, I can simplify it by saying, well, once you've told me about those risk factors, I never have to ask you about them again, because you don't not have diabetes. You might have better controlled diabetes, and there aren't that many people, frankly, that quit smoking. And once you're a current smoker, most people are going to stay that way. So it's probable that you could extract them out of the electronic health record. And if you can do that, it's an algorithm, and the lab result can come back through automated means like any commercial lab can come back. And so you could basically order this from your EHR, extract all the risk factors, and then give people back the result in their EHR. The interesting part about all of this, and with your implementation science hat, the part that might intrigue you and certainly intrigues me, what are then clinicians going to do with the information? How do you make it actionable? You know, rheumatologists, honestly, are not too keen on managing cardiovascular risk. They're perfectly happy and delighted to manage, you know, less than well-controlled rheumatoid arthritis, but not so much about measuring statins or PCSK9, et cetera. So, you know, who the responsible physician is or provider is to act on the information, that's quite critical. But I think you could automate it, most of this from a data and an analytics perspective, to provide this information really at the point of care.
While being in primary care, I can tell you right now that's very much of interest to primary care clinicians. And I think that one of the things that's often lacking is there are a lot of medications that have an increased cardiovascular risk, even though that's not their primary target, antidepressants, for example, in patients with obesity. So I did not catch this on, I thought I saw something on your poster, which you've now taken down. Did you look at weight status in your subgroups? And that's just a 100% personal interest question.
We didn't look at weight explicitly, but in this poster, because we know from prior work, because we actually are measuring multiple adipokines, those adipokines outperform body mass index or probably anything except for, you know, a research grade bone density test that measures body composition. So if you measure adiponectin and leptin and resistin, you're going to do a whole lot better, whatever you're studying, in my opinion, than simply measuring weight or BMI. So, and that's been looked at formally before. So the short answer, no, but it's because we don't need to. We're measuring multiple adipokines.
Fantastic. Very happy to see that. Leah, anything from you or anybody else who's on the line?
I was going to see if any other participants had a question for Jeff. Jeff, real quick before, and kind of, want to keep us rolling, but when you, just a question about your tokenization, how well did that work? And do you have a sense of, I guess, what was your experience? I've only recently been, have dealt with that for a couple of studies, but didn't know if you had any feedback, if it worked well.
I think the maximum trust but verify will serve you well with tokenization. When we link Medicare data, we're linking patient level data that is frankly identifiable because it can be, and it's considered research identifiable. When you subject your data and your personal identifying information or your PHI to somebody's tokenization algorithm, that's the trust but verify that I'm advocating. We intentionally said, we're going to dial up the sensitivity because we don't want, to lose anybody that might match, because then we're going to do some internal quality control checks to make sure that we really have the people that we think that we should have. And so we will do additional work to improve specificity. My sense is that the accuracy is probably 90 to 95% with that approach, but I think you would be well served to do what we did, which is to say, let's look at something that can overlap between these two data sets that we're joining, just sort of as a checksum or a verification step to make sure that you have what you think you have. You can't do it on identifiable data, obviously, otherwise why tokenize, but you should almost certainly be able to do it on things that are in common between both data sets. In this case, what's in common between both data sets is you generate a billing claim for the biomarker, and if you have the lab's data, when that biomarker was ordered or when it was collected. So you can actually anchor or link on the date of the test being performed and the date that it was billed for being performed in the billing data. So that's what I'm calling this verification or this checksum to make sure that the people who you think you've linked are actually the right people, because you absolutely do not want false positive people linked. You need to link the right people. So that additional specificity step, I think, is really critical, because 5 to 8% of people probably are going to be mislinked. You can do it all in one step, and that's fine. You'll just sacrifice some sensitivity on the front end.
Sure. No, very good points. I appreciate it, because I think those of us who work in genetics and pharmacogenetics is something that we're starting to face more and more with the availability of different data sources. So thank you. I guess we should move on to our next speaker, unless there's any more questions for Jeffrey. Don't see any.
Okay.
Thank you, Jeffrey. Really
Thank you very much.
Appreciate
I'm
it.
loving how these presentations are getting us closer to actionable clinical reality. Very, very helpful and interesting.
So next up, is it Guilherme?
Yes, it's a Guy. You can call me Guy.
Guy, welcome.
Yeah, thank you. Let me share my screen.
Yeah. Turn it over to.
Can you see my screen?
Yes, we can.
All right. So Leah, Gideon, thank you, and thank you everyone. My name is Guy Lopez, and I'm currently a senior research scientist at Premier. But at the time I was working at the Mayo Clinic as a research associate, I had the opportunity to lead this study that's called Identification of Sex-Specific Genetic Associations with response to opioid analgesics. So opioids affect individuals in different ways, right? So some people, they really benefit from opioids, while others, they suffer from terrible adverse reactions. So for example, we know that biological sex affects response to opioids. Women are more likely to experience adverse reactions than men, right? We also know that a variation in genes that are involved in opioid metabolism or in pain perception, for example, they can also affect how we respond to opioids. But we don't know yet whether or how these factors interact, right? So here we investigated whether there are sex-specific associations between generic variation and opioid response. So our study included a bit more than 7,000 white patients who were prescribed at least one of the 4 most common opioids in the United States between 2005 and 2017. And the 4 opioids were codeine, tramadol, oxycodone, and hydrocodone. These patients were part of the RIGHT-10K study, which is the largest preemptive pharmacogenomic study in the world, conducted at the Mayo Clinic. We extracted 2 outcomes from the patient's electronic health records, which were poor pain control and adverse reactions. And for the analysis, we created 2 independent subcohorts. One was included the patients prescribed hydrocodone and or oxycodone, and the other one, those prescribed codeine and or tramadol. And we also conducted, I'm sorry, we also conducted 2 main analyses, one at the variant level and one at the gene level. So as you can imagine, right, the results were very extensive. So here I'm going to report only a snapshot of what we thought were the most important results. So if you look at the figure 1 here, this is the results of the variant level analysis for the outcome poor pain control among patients prescribed codeine and or tramadol. And these are Manhattan plots, right, for men and for women. And we use 2 significance thresholds, the p-value and the q-value. And based on this, you can see that we found 2 variants associated with poor pain control in men, but not in women. And they are both in the CYP1B1 gene. Moving on to the gene level analysis, here at figure 2, we'll focus here on the adverse reactions outcome. And genes with significant association with adverse reactions, they need to be in this quadrant here, right? It's a little tiny, but it needs to be above and to the right of those 2 lines. So what we can see here is that in both drug groups, we see that the FAAH gene was associated with adverse reactions. When we stratify by sex, however, we see that this gene remained associated in men, but not in women. Right. So in this study, we identified sex-specific associations between genetic variation and opioid response. But beyond any specific results, right, beyond those points, I think that the value of our study relies on showing that association studies should consider sex as a biological variable in their analysis. Very rarely, we account for sex in such studies. But here is one example in which results differ by sex. The whys underlying our results are unclear at this point, but we hope that this study will help push further research on sex differences in this field. Thank you.
Thank you very much. That is really interesting. My summer reading was it's something called Invisible Women, and it's data biases introduced based on gender. And one of the areas that they explored was medications that might be more effective or may have gotten to market if they had been analyzed separately in men versus women. So your topic is very pertinent and timely, and we hope to see more of these. So
I do agree with you. Yeah. So the NIH has a push for incorporating the variable sex, right, in analysis. And at the Mayo Clinic, I observed the same push, right? So we had internal funding for sex differences research, and I think this is just a great example, right? Of typically, we don't consider sex in the analysis and we wouldn't see those differences. But then when we incorporate sex, then we see, wait a minute, men and women, they respond differently, right?
Mhm.
Yep.
And it's having sufficient numbers to do it stratified, right?
Mhm,
Because
mhm.
depending on their reaction, it can actually, when you do it on an average, it can negate the effect completely. And so this is what was very interesting about your results.
Yep, sure. Mhm.
And when I give lectures on this, I do usually mention and say, apologize to the men and say, we literally have twice the guts you have as a male. So, the whole hormonal. So this then, of course, in your research, I mean, you're doing the genetics, but it would be very interesting. One of the other things in pharmacoepidemiology that we need to consider is if you're doing drug effectiveness, safety, whatever type of research in women, even if you're adjusting for the genetics, which should be the proper way of doing it, the hormonal cycle can affect
yes,
efficacy as well, and dose. And so there's
yeah,
lots of work there to be done. So
that's true. Here we were not able to control for that, right? So most women were like above 60, and but yeah, maybe another, like in the future, like future research should account for that. The other thing we didn't, we were not able to incorporate in our study was dosage, right? So basically we go back to the medical notes, right, to the EHR.
yes.
Now, what was the exact dosage they were taking, and were they really taking the way they were supposed to, right? So
Well, the dosage would be important because in the work that I do and what I look at is that you do find gendered, so not biological, but gendered response in dosage. So
mhm.
young women tend to be under medicated when it comes to pain control.
Oh, wow.
So
Wow.
yeah, so this is you've got a whole research program you can
I
pursue
do.
here.
I do.
This is
Well,
really fascinating.
yeah. So I'm writing down all those ideas. And the book you read was called Invisible Women.
Invisible Women. It's actually written by a health economist or by an economist, and one of the things the reason why they brought this up is that with, I think somebody else mentioned artificial intelligence and machine learning, is that if you have a bias in how you collect data and you don't do sex stratified analysis, you can amplify existing biases by almost 50% with using artificial intelligence and machine learning. So anybody who's interested in drug safety, effectiveness, and development, including the genetics component, should be looking at this a lot more carefully. And, it's already one thing to just create two piles of men and women, but then you also, in the female pile, there is all sorts of other things that can be happening that
Subpiles.
impacts.
Yeah.
Yeah, there's
Yeah.
a few subpiles. And I noticed that you had race, and that also for men and women have differential impacts. And
That is
actually, if
true.
you are a young Black or Latina woman, you're really going to be under medicated for pain control.
Yeah. Yeah.
So
Okay.
I'll
Yeah. I
I'll get off my
appreciate it. I
soapbox.
appreciate all
I
those
loved
comments.
your poster.
Yeah. Thank you.
Leah, do you have a question that's not me ranting about gender and sex differences?
No, very wonderful poster, very straightforward. So appreciate that. I don't see any questions in the chat.
Yep. Thank you, Leah.
Yeah, thanks so much. We really appreciate it. So maybe we'll move on to our last speaker, interest of time. I think Jessica. And if anybody has questions, feel free to populate in the chat. All right, Jessica, I'll turn it over to you.
All right, let me the right thing shared here. Okay, can you see this? Okay.
Although we still also see all your individual slides on the side. Jessica, I don't know if it's just me, but I can't hear you. We lost your sound. No sound.
Jessica?
I can't. Let me message her.
Oh, no, she thinks she's giving her presentation. This is awful. I'm gonna do some sign language here to Jessica. I don't know if she can see me.
She should be able to see you if she has you up somewhere.
Okay. She might not, because sometimes when you put your full screen on, you
Yeah,
can't
you
see.
can't see.
You can't see the chat, or you can't see the yeah, you can't see that there's a chat or that we're waving, and oh, that's a bit unfortunate. Well, at some point she hopefully will stop, and oh, there she's gone mute. Jessica? Oh,
We can't hear you.
she may have to sign out and sign back in. Oops.
We can't. Amy,
I'm gonna
Can't
take
hear-
her. I'm
We
gonna take
can't
her
hear-
screen sharing away, and maybe
take her
that
screen
will.
sharing away, and then she'll be able to see us. Yeah,
Yeah.
thank you.
Jessica, can you hear us at all? We,
No, she doesn't.
we see you talking, but we can't hear you.
Let's
No
see.
sound.
All right, that's not a
Poor
real
Jess.
thing.
All right.
All right, Jessica, maybe leave the meeting and come back in.
The good news is we have a little extra time for this. Technical difficulties. Sorry, everyone.
Oh, it could be her AirPods. Yeah.
Yeah.
Nope, it wasn't the AirPods. I'm gonna just send her a message.
I sent her a chat. I don't know if she's on. She's trying.
So while Jessica is attempting to sort out her technical difficulties, just to let everybody know that we will be having a SIG meeting on September 28th at noon EST. So we'll put that up on ISP Connect, as well as circulating an email if you volunteer, if you've written in to be part of the SIG community, but you can find us on ISP Connect.
Yep, and I'll be sending a notice and Zoom link via the exchange. But for those of you on the call, if you're interested in joining the SIG, please also reach out to me and Jill. And for any of the presenters today, if you'd love to present to the larger SIG, I know it's late for a lot of the Europeans, so but we would love to, have you present in a little more detail your work that you're doing. We find they're very interactive and usually spawn some new ideas and questions as well.
Jessica, can you hear us? Oh, it's not looking good. Thank you, everybody, for your patience. Please hang in a few more minutes. I'm sure we'll get this sorted out and we can hear Jessica's presentation. Oh, by the way, I will just plug the, I am moderating a session that is got an EDI mandate. I actually should look at when that is. So equity, diversity, and inclusiveness in research. Looks like there's some real fascinating sessions there. I think it's tomorrow. Yeah, it's tomorrow at 11:30 Eastern. And it's the impact of studies in STMA on acts and utilization. So that's sort of one of my areas I'm very, very interested in. So for those of you who are still hanging around for the rest of ISB, or ICB, as they call the conference. Jessica, how are you doing? Can you hear us? We can see your lips moving. If you can hear us,
hear me?
yes, you're on the phone line.
Are you
We hear
kidding?
you.
Oh, wow. All right, and I can hear you. Sorry about that. I don't know what happened. We were all good, and when we started, okay. So you heard none of what I said. Is that?
No, we didn't. So can you start? We'll consider that a little rehearsal for you. And why don't you start again?
Oh, fun. Okay, let me back up. So are my slides still shared?
No,
No, I need to
Amy had
reshare
taken
them.
you off so that we could get your attention. So Amy,
No,
if you
it's
could
okay.
make,
So yeah, let me try again. That's when I lost you guys when I shared my slides. So hopefully this still works. Are you still with me?
yep.
Okay. All right. We'll try this again, and I will be a little bit brief. So essentially, I said, I wanted to thank my colleagues who are my co-authors, Carla Vossen and Tara Isherwood. So as way of background, personalized medicine in the United States is increasingly a higher proportion of drugs that are approved. Currently, 1 in every 4 drugs under the FDA was approved as a personalized medicine. And there's a variety of regulatory tools that are used to accelerate the market launch that are often accompanied by 1 or more post-marketing requirements to collect additional data as far as safety or efficacy or optimal use. And so we wanted to look into that a little bit further and see what kinds of categorizations there might be and correlations between regulatory tools and post-marketing requirements. And so we looked at data that was approved by the FDA between 2017 and 2019. All right, so we use publicly available data sources to analyze and describe the information. So we've got a list of resources references at the end of the presentation for those individual resources that we utilized. Okay, so back to results. So 56 personalized medicines, including 4 gene therapies, were approved during the period that we had under investigation from 2017 to 2019. Most frequently, the indications were around hematological malignancies and congenital familial and genetic disorders. All right, so for 53 or about 95% of the approved medicines, they required greater than or equal to 1. Yeah.
Sorry to interrupt you. Your slides are not advancing. Okay, so you're still on your main slide, your title slide?
Well, they're advancing on my view, but you're not seeing them. Okay, you know what? Let me just.
I'm really sorry to interrupt you mid-presentation.
No, it's okay. We won't have to worry about it. So let me just go to the poster. Boy, oh boy, this is the most technically challenged.
We see the poster, so you're good there.
Okay. All right. You got it good? Okay. Hope it's big enough to see well. Okay. So results then. So we looked at the type of indications that the medications were approved under, and then we looked at the various regulatory tools that were used. So as I was saying, about 95% of the approved medications required at least one or had at least one regulatory tool that was utilized, and then 11 were approved under accelerated approval pathways that had at least 2 or more regulatory tools that were utilized as well. So the types of regulatory tools were broken down into breakthrough designations, priority review, fast track, and orphan drug designations. Okay, and then so there were another 45 medications that were not approved through any kind of accelerated pathway, and the majority of those used 3 or 4 different regulatory tools. Everything from no tools to single regulatory tools, but the majority had multiple ones that were utilized. So then the main findings, about 91% of the personalized medicines had at least one post-marketing requirement that was mandated to be conducted by the FDA. There were new clinical studies required for 36 or 64% of the medications. So when we looked at these by whether or not they had been approved under the accelerated pathway, there were some differences that we noted in the types of regulatory requirements that were required. So for the 11 that were approved under the accelerated pathway, the primary types of studies that were requested were pharmacokinetic in nature, efficacy studies, and pediatrics. Whereas for the 45 that were approved and did not follow the accelerated pathway, they focused more on safety studies, pharmacokinetic, and pregnancy studies. So there were some differences between those 2 categories of pathways. Okay, and then in conclusion, between 2017 and 2019, so there were a total of 56 approved medications. So again, primarily in oncology and gene therapies. Most use at least one regulatory pathway, and a good portion, 20%, being under the accelerated pathway. So while both groups had approximately 64% of study or approved drugs that required a post-marketing study, additional clinical studies were distributed differently versus the focus on the safety versus the efficacy. So in the future, we would like to look at similar types of breakdowns for other regulatory bodies and regions of the world. So kind of, I think, a next step for us will be to look at EMA approvals and see how that compares with the types of post-marketing requirements and mandates are made for drugs in those regions of the world. And then also, looking at some of the design metrics for the individual types of studies to see if there's any trends around, the types of outcomes or study designs, et cetera, that are utilized to fulfill those post-marketing requirements. Okay, so any questions for me?
So thank you for that. I'm just wondering, I'm curious, like you've talked about what was needed and what was what the post-marketing requirements are, of course, as a pharmacoepidemiology that exclusively does post-marketing studies. I'm just wondering, do you know, I don't know if you said this, and if you did, I apologize, but what's the follow-up? How do they know the studies have been done? So because this seems like a lot. This seems like a very big number that need additional information.
Yeah,
I mean, that in and of itself is fascinating, but.
Yeah, no, certainly. So the FDA certainly does track, and I think, and I don't work at a pharmaceutical company, but certainly, you know, they're very keenly aware. I mean, every study has a timeline that it's intended to be met by. That's agreed to upfront with the FDA as sort of when they're going to launch the study, when they're going to conduct their interim analyses, you know, have, you know, their final analytic outputs that are reported to the FDA. And certainly, you know, at least from a safety perspective, they are monitoring those typically throughout the course of the study, making sure they're on track as far as enrollment goals, et cetera. And so in some circumstances, it's, you know, complete the study and, you know, they agree on the protocol and methodology with the agencies. And sometimes, you know, it's more about, you know, have they met any sample size requirements and in order to get released from the post-marketing requirement. So it's something certainly that the agencies focus on very carefully, and they follow and they monitor to make sure that their obligations are met.
Okay,
So no
this
small
is fascinating to me. I'm always, because I've done a bit of research where we look at what was funded versus what people published. And that's always really fascinating. Sammy has a question for you. I'll just read it out from the chat and said,
Sure.
well, 2 questions. Was the definition of personalized medicine in the case based on the use of a companion diagnostic in the indicated label? You can take a look in the chat if I was.
Yeah, no, that's okay. So yes, we followed the FDA designations as far as, you know, the types of, you know, categorizations that were used. So in their reporting documents, they will actually, you know, categorize the various regulatory tools. So if you go in and look at some of those resources that are in the links with the references, it will highlight them for you. So we've looked at, you know, a variety of different types, but for the most part, I think as far as, you know, definitions from FDA with regard to, you know, accelerated, you know, approval, you know, it's based on using a surrogate endpoint, and so they've got, you know, different elements that they use as their definitions for those various regulatory tools and when they're applicable. And I think that's something, you know, that each drug developer probably works with the FDA on agreement on the submission pathway.
Okay, and Sammy had another question for you as to whether your team got a sense of whether personalized medicines made greater use of FDA regulatory tools than approved medicines that were not personalized.
That's a good question. So we didn't really compare it to non-personalized medicines to see how it differed. My guess is that because the populations to treat are going to be smaller, that, you know, you would expect that to be the case. But that's something that we hadn't looked at, so it's a good thought to follow up.
Okay, Leah, any other? I don't see any other questions in the chat.
No,
Donna has posted.
she put a resource in there for folks if they want to look at that link to the FDA as well for some of the questions that came up. Thanks, Donna.
Anything else, Leah? That was very interesting.
I don't have any other questions, and I know we're just about at time. So I want to thank everyone for joining. I know it's late after a long day of virtual meeting and sitting. So appreciate it. Big thanks to all the presenters for submitting your abstracts this year. I know it was hard. A lot of people have virtual conference fatigue. So thank you for that. Thank you for joining. Please do reach out to me and Jill or look out for some invites for the SIG. We'd love to have you join and also present some additional data, and wish you all the best for the very end of the conference coming up tomorrow.
Thank you, everybody.
Thank you all.
Thank you.
Thank
Bye-bye.
you, Amy, for your help in facilitating the virtual poster walk. The poster walks are always a highlight for our SIG, so we really appreciate that.
My pleasure. Thank you all.
Take care. Bye-bye.
Yep,
Bye-bye.
Thank you. Bye.